I'm at ASH, where I have presented the result of the Effikir trial. The Effikir trial is a phase 2 randomized placebo-controlled study of Lirilumab, a monoclonal antibody directed against the KIR receptor in elderly patients with AML in first complete remission. The objective of the trial was to demonstrate that Lirilumab is able to improve the leukemia-free survival of elderly patients in the maintenance situation. Lirilumab is an anti-KIR monoclonal antibody. The mechanism of action is a blockade of the KIR inhibitory receptors presented by the NK cells in order to activate, to modify the balance between inhibition and activation of the NK cell and to activate the anti-tumor activity of NK cell. So the eligible patients were elderly patients, older than 60, in first complete remission of AML. These patients had to have received 1 or 2 cycles of consolidation before inclusion. Once included in the trial, the patients were randomized into 3 different arms. In the first arm, the patients received Lirilumab at a dose of 1 milligram per kilo, a dose which is predicted to provide a full KIR saturation, a continuous full KIR saturation for the whole treatment period, which was planned to be 24 months. In the second arm, the patients were scheduled to receive Lirilumab at a dose of 0.1 milligram per kilo every 12 weeks. So that this is a dose which is predicted to give a discontinuous KIR saturation. And the third arm was the placebo control arm. 150 patients have been included in the trial, approximately 50% for each arm. There were no major imbalance in terms of patient or disease characteristics at inclusion between the different treatment arms. Approximately 30%, 35% of the patients in the placebo arm and 0.1 milligram per kilo arm received the 24 months of the planned treatment with Lirilumab, while only 6% of the patients in the 1 milligram per kilo received the full treatment. The reason of that was that there was an excess of relapse in the 1 milligram per kilo arm. And a decision of the DSMB during the trial was to stop earlier the treatment for the patients in this arm. The major reason for discontinuation for all patients was relapse. The results of the study are negative because the improvement of LFS, which was the endpoint of the study, has not been reached. There was no significant differences between the treatment arms and the placebo control arm. In the placebo control, the median LFS was 13 months, as expected. It was 17 months in the 0.1 milligram per kilo arm, but no, it's not significant. And it was only 6.7 months in the 1 milligram per kilo arm. So there are reverse trends between the treatment arms in that study. These results were confirmed in terms of overall survival, and subgroup analysis didn't find significant interaction between the patient characteristics or the treatment results. The safety was fine. There were no unexpected adverse events in the trial. So in conclusion, the study failed to show that Lirilumab is able to improve the leukemia-free survival of elderly patients in first complete remission of AML. Interestingly, there was this reverse trend between the results in the 0.1 milligram per kilo arm and the 1 milligram per kilo arm, suggesting that the dose schedule and the continuous versus intermittent KIR saturation may have some importance. There are just hypotheses because we are not sure that some confounding factors could have modified the results of the study. We are currently conducting biological studies and immunological studies to look, for example, at the NK cell status at inclusion and look for potential confounding factors which may have modified the results. Clearly, from this data, there is the further development of Lirilumab in this setting, the maintenance of AML in the elderly will not be pursued. There is a room for further study of Lirilumab, probably to optimize the dose schedule, but also to look at the benefit of combining Lirilumab with other treatment options, because it's possible that the release of the inhibition of the NK cell is not sufficient to have a clinical impact, and that it's necessary to have other activation of these immune effectors.